Thanks very much, Jinju. And it's a great pleasure to be here, and especially to share the stage with Jinju, who was in my laboratory for a good period of time, and we had a great time while she was there, and we're very productive. And I was very appreciated her as a scientist, as a thinker, and a very apt and technically adept scientist as well. So today I'm going to talk a little bit about modeling aging, and specifically about neurodegenerative diseases, and talk about some of our recent findings. I'm also, at the very end, I'll give a little preview of some new data about how we move from two-dimensional modeling of induced neurons into organoids, and our attempt to fill out the organoids in a more complete manner. So I'll get to that at the end if we have time. So let's see here. So aging is the progressive decline of fitness with time. And as a result of age, there's an accumulation of damage and likely a programmed cellular response. In other words, it's likely that age is built into the systems. We don't know whether or not this is actually something that is pre-programmed or it's wear and tear that results in the deterioration with age. And it's a very hot topic of investigation. There are currently, as you can see on the left, a lot of things that happen with aging: genome instability, telomere attrition, epigenetic loss, drift, mitochondrial function, senescence. And if you go to an aging meeting, you often find that there are sessions which deal with each one of these individually. And our focus is beginning to be less about treating each one individually and more about looking at the interface between them, thinking of age and the consequences of aging in terms of their interactions between these systems. So what we do know, though, for sure, is that age is the largest risk for many diseases, not just neurodegenerative, but also psychiatric disease and cancer and diabetes. So I will focus on or make a lot of the comparisons between age-related degeneration and Alzheimer's disease. And we've shown previously that Alzheimer's disease is not just an extension of aging, but it is a disease itself. And I think that's an important distinction to be made. Now, as it stands now, although this hypothesis is being revised, has been sort of the amyloid hypothesis, where the tau tangles and amyloid plaques that exist in postmortem tissue clearly exist and are purely correlated with the disease, but it has been considered or hypothesized that it's causal or has been the initiating event. And it seems less likely, given the last 10 years of 1,000 trials and $24 billion that have been spent on clinical trials that have failed, that this hypothesis needs to be revised and or elaborated. That being said, a lot of model systems have been used, but most of them are taking wild-type animals and overexpressing human mutations with the idea that they'd understand the disease. Now, it's not unreasonable to go in that direction, but as we're beginning to learn and appreciate even more all the time, is genetic background is a very important feature of any mutation that occurs. So it's not just the mutation itself, it's the background on which the mutation occurs that's important. We've moved to looking at human cells and taking into consideration the human genetic background as being an important feature. And with the discovery of the Yamanaka factors and the ability to reprogram, we've taken advantage of this. So initially, we took fibroblasts from patients and made fibroblasts and reprogram them to iPS cells. And when doing this, you can get derive cells of many different lineages, cardiac, retinal, pancreatic. Really, it's amazing the number of cells that can be authentically be derived, including neurons. We did this study with 20 patients that ranged of age from 1 to 80 years old, so fibroblasts were young to old. We took primary fibroblasts and did RNA-seq, and we looked at the differences. We actually, using fibroblasts, we could find age-related disease, age-related genes that declined with age. Not a lot, but we did see some. And these were with early computational tools that have been advanced further since. However, when we looked at the iPS cells from the same isogenic patients, we found essentially no genes different between them. And then we took and made neurons from these iPS cells. We also found very little or no differences between with age, change with age. Well, what that means merely is that the iPS cells did their job. They reprogrammed, they erased the epigenetic program and turned them back into embryonic cells. And as we know, Alzheimer's disease, aging, or Alzheimer's disease really requires these age as a feature to be able to pick it. We've lost aging in this model. So we've turned to another model where we can use two genes, directly reprogram fibroblasts, NG2 and ASCII, which are small, which are pioneer transcription factors. And more recently, we've identified some small molecules that facilitate this direct induction. And these pioneer factors open chromatin and allow for specific transcription factors to bind and induce a direct conversion of the cells. And this is just an area still of major investigation of trying to figure out exactly how these factors do it. But it's clear that they're opening chromatin for transcription factors to bind specific sites. But quite remarkably, in a 3-week period of time, we go from fibroblasts into this network of neurons. Now, when we look at the transcriptional differences between young and old, or across young and old, we now can pick up a number of genes that are significantly different. So we've preserved this aging phenotype, including one gene that we've spent a lot of time on, which is RAMBP17. It's a nuclear import gene that controls the transport of molecules from the outside of the nucleus to the inside of the nucleus, and is downregulated with age. And we developed an assay where we could monitor the nuclear import and export signaling ratio. And what we see is this nuclear import export is disrupted with age as a function of this gene. Now, the good news is that it's not a mutation in RAMBP that occurs with age, but rather an active downregulation, because when we make iPS cells out of very, very old fibroblasts that have that the fibroblasts also show defects in RAMBP17, we can reverse it by inducing iPS cells. So that's a reversible aging phenomenon. We're now continuing to work with this to see if we can find small molecules that can repair this nuclear import defect. So since we've been doing these studies, we put all these vectors on Adgene and sent them out to many people. And a lot of people now have shown that there are epigenetic changes, methylation, neural survival, age-related changes that can be picked up now using this strategy that have not been previously found. Importantly, we think that aging, it reveals that aging is not just a single phenomenon, but certainly neuronal aging involves a variety of mechanisms. Here's one that we have done in collaboration with Steve Horvath, who developed the DNA methylation clock. What you can see down here is this is chronological age here, and this is DNA methylation age here. And for iPS, human iPS cells, NPCs, and neurons, they are all chronologically aged around birth. So they're very, very immature. Alternatively, the IN fibroblasts and INs, if you look at their methylation clocks, they're all proportional to the age from which the age of the patient the fibroblasts were taken from. We can do this another way, which is to take the RNA sequencing clusters from brain span, which are a set of genes that are expressed at various times prenatally and postnatally. And what you can do with this very useful tool is take your RNA-seq and feed it into brain span, and it tells you how old your cells are. So what we find, for example, is if your cells were taken at 8-week, you can feed your cells and it'll tell you if they're young or old. And when we do this, what we find is that for the iPS cells, prenatally, they compare to prenatal, but they're low comparison similarity scores with age. And it's just reversed with our INs, where they're lower similarity scores prenatally and they get higher postnatally. Again, suggesting that they preserve their aging fate. So let me get into the meat of this, where we talk about our comparison between age-matched controls and our AD patients. A reasonable sized cohort of 19 controls and 16 AD, 13 sporadic, and 3 familial forms. So this just gives you a summary of the fact that all the fibroblast cells, here's what they look like normally with fibroblasts, and they're Vimentin positive. And then within 13 days, they've converted into neurons. And independent if they're controls or ADs, they're mostly neuin positive, V-glute, so they're excitatory, and a small 10 to 15% are GABAergic. None of the controls or ADs show any difference in their 42 to 40 secretion, the ratio of those peptides, except for the FAD or the familial forms do show those differences. So sporadics don't spontaneously show any difference in the A beta. So we did RNA-seq for all of these patients. And when we look at the fibroblasts from the control versus AD, we see perhaps as expected, but we were sort of hoping we might find some signature differentiating AD from controls. We don't see any difference there. However, when we made IN cells, we have massive numbers of cells that differentiate between the age-matched controls and AD. When we then do a differential expression mapping of these, what we see, if you control for sex, age, ApoE, and diagnosis, as well as FAD mutation, they separate really quite nicely. In fact, some of these controls here that were initially listed as controls were from patients in the Alzheimer's disease clinic at UCSD have converted into AD. So we were able to pick them up as ADs early on, even though they were suggested to be controls. Now, how well does this RNA-seq match with the existing postmortem data? So we downloaded data from RNA-seq from Alzheimer's disease over the last 10 years, actually 20 years if you look at it. And what you get is these are the correlations, positive correlation, negative correlations, and they match very nicely with our RNA-seq data, so they're positively correlated, all except for this one group in 2013 of the parietal lobe. There was a poor correlation with those, but otherwise, there's a very good correlation between the two. There's also a recent, not so recent, but recent single cell sequencing by Grubman et al. In Nature Neurosciences. So we downloaded that data, comparing their RNA-seq with ours. And of course, ours are IN, so they're only neurons. So we correlate ours with the neurons for differential express overlap, exclusive DNA expression, linear regression, all very, very highly correlated between our data and this single cell published data. All in all, we feel like this is a pretty good, that our IN is a good model for capturing the defects. Now let's see what the defects are. So again, looking at sort of Go terms or enrichment terms, we see that if you look at those that are downregulated in AD relative to the age-matched controls, you get terms like rest, synaptic LTP, synaptic transmission, synaptic plasticity. So all those features which are involved in the cell identity, the function of a neuron, are downregulated in Alzheimer's disease. It's almost like they're de-differentiating into an immature state or a quiescent state. However, on the other, this was all RNA-seq, so we wanted to get a confirmation of this using functional assays. So we took our INs, stained for synapse and PSD95. These are synaptic markers for pre and postsynaptic sides, so we can see whether or not they're in apposition to each other. And we certainly see a decrease in AD in the synaptic connectivity, morphological connectivity in these cultures. In addition, we did morphological complexity analysis, looking at the complexity of the branching, and we see a consistent decrease in complexity of branching in the AD neurons. And we do calcium imaging, and we see decreases in the calcium activity of these cells in the AD group versus the controls. Now, what is upregulated? We saw that the cells are de-differentiating or losing their neuronal identity. But what's happening with the upregulation? Quite remarkably, the genes that are going up have hallmarks of what maybe not so surprising of cellular stress, increase in apoptosis, but there's also a strong inflammation signal and also a signal of many cancer-related genes like oncogene signaling, mixed signaling, cell proliferation, YAP-TZ or Hippo pathways and Notch sequences all upregulated. Very strange cluster of genes are being upregulated in these Alzheimer's versus the controls. So what's the connection? Well, it seems like there's an over-representation of cancer-like signaling pathways. So you pool that together with what's going on with the downregulated genes, it appears what happens is the cells are shifting back into a de-differentiated state where they lose their neuronal function and fall into a dysregulated cancer-like signaling. And I'll get into this in much more detail in the second part of my talk. But let's see what else is going on here. And if we look at DNA damage pathways, so these are genes that are involved both in DNA repair and DNA damage, we see a dramatic increase in DNA damage-related genes and suggesting that there's genomic instability. In addition, we see an increase in reactive oxygen species, where there are increases, and I'll show you in a minute, effects on DNA. And it looks when there's an increase in total DNA content within the AD patients, which is like patient neurons, which is likely similar to this idea that they're attempting to go back and cycle. And we saw that in the RNA-seq data where there was a signal for proliferation. So let's take a look a little bit deeper into the functionality of some of these measures. So we took our fibroblasts, converted them into INs. Sure enough, we see that increase in ROS. And we see when we use a staining for H2AX, we see an increase in nuclear staining for this DNA repair molecule in the ADs compared to the controls. So nuclear DNA repair is indexed in these cells. And as I showed you, was an increase in the DNA content within these cells as well, in the age. Now, what about this cell cycle? How deeply into the cell cycle do they actually go? We've looked at cyclin B1 in the IN neurons, and we definitely see an increase there. And you can see the nuclear localization of this protein there. So do the cells actually go into cycle? So we labeled the cells for a period of time for 72 hours with EDU to see if they actually would fully go into cell cycle. And we never saw a dividing cell. So they're converting back a ways into cell cycle, G0, G1, but they're not fully going through mitosis. So it's an attempt, as I'll try to convince you a little bit later, is we think that they're reaching back into their metabolic pathway to look for bulk-like molecules to assist in the repair process. So let's look at the cell fate instability or the epigenetic identity of these cells. So to do this, we used KAIX, and you all know that generally the way we think about it is that in progenitor cells and stem cells, the chromatin is a little more open, available for transcription factors to bind and define who the cells are going to be. So open chromatin is a hallmark of an immature cell. And when we look at KAIX in our AD versus controls, we see a significantly higher number of peaks in the AD versus controls. And there's a specific differential upregulation of accessibility of chromatin within AD versus controls. And where we look where that exists, there's a very much of an increase in availability at the promoter targeted start site. So open promoter target start sites regions are associated with cancer, metabolic, and apoptotic genes when we look at the GO terminologies. These are the things that we're seeing opened up. Very consistent with what we were seeing with the RNA expression, these metabolic pathways and cancer-related genes. If we do motif mapping, we can find out very specifically which genes are showing this open chromatin. And again, these two pathways that we're particularly interested in, these metabolic pathways for glycolysis, LDHA and H2K, both critical pathways for glycolysis, as opposed to oxidative phosphorylation, are upregulated. You can see this much more open in AD versus controls. And then many of these cancer genes, RUNX1, HIF1 alpha, STAT3, all showing more open chromatin in Alzheimer's disease relative to control. So DNA binding motifs for stem cells and cancer transcription factors are more accessible. So are they for Alzheimer's disease. To end up this portion, I want to ask the question, is age a crucial component to be able to reveal these differences? So how can we do that? How can we ask that question? So we have two ways of generating neurons. One is the direct program, which gives us induced neurons and captures aging. The other is we can take isogenic cells, these same fibroblasts, make iPS cells out of them and make neurons. And then we can compare the transcription of these isogenic cells from iPS derived versus IN derived. Yeah. So the good news is when we convert these fibroblasts from this whole cohort using the reprogramming factors OSKM to make our iPS cells, we can use the same vector that we used from fibroblasted neurons from iPS cells to neurons. That way we have controlled for not just the background genetics, but also the converting genetics. The only difference is that these are young and these are old. Now we have neurons that we harvest and we do comparisons between neurons we're looking at the difference between controls versus Alzheimer's disease neurons derived from iPS cells versus derived from IN cells. So not surprisingly, in the iPS cells alone, we see no differences. That's not a big surprise. But what is a little bit surprising is the iPS-derived IN neurons show very little differences between them, unlike what we saw with the INs that I just spent 20 minutes telling you about. And when you look at the differentially expressed genes, we find essentially no significantly differentially expressed genes in the iPS-derived neurons comparing age from controls. So I'd like to make the argument that age is a crucial factor in being able to detect aging. It's a major risk factor, and all the genetic underpinnings can't be revealed until they're later. I'd also like to highlight the fact that what I've showed you is that it isn't one of these events that's important, but rather aging revealed metabolic problems, DNA problems, nuclear import problems, and it's really a combination of these. I think we think of brain aging and disease as really a homeostatic system, which is controlled by a variety of systems, all of which are compensating for each other in normal aging, but a secondary stress results in cognitive decline. And that sort of description should give you a hint to this idea of cancer, where you have age-related decline or age-related changes that occur with cancer, but a second hit. And you can compensate with, even if you have a genetic background for cancer, but if you have a second hit, which makes you more vulnerable, then you can have cancer, just like we believe you can elicit cognitive decline. All right, so let's go a little bit deeper and try to analyze more carefully and more systematically the changes that are occurring in age in not just our cells, but in postmortem tissue. So we used WGCNA to find clusters of genes from our transcription from the AD patient INs, and we pulled out clusters, or we call AD modules, that are clustering around, that give an insight into those events or clusters of genes that are involved in upregulation or downregulation of the cognitive decline that occurs with aging. And we did the same thing for postmortem tissue. So we took existing data from postmortem tissue of Alzheimer's disease, performed the same WGCNA, and what we find in both cases is a standout motifs that emerge is a metabolic shift to glycolytic reprogramming is indicated both in the IN and from this ROS map, or the religious orders data set that you can get from the University of Chicago and Rush. This data is openly available for everybody to interrogate. Let's see here. So we take it one step further using a stepwise attempt to isolate those genes that are best characterized when you're comparing now the INs and the ROS map data. And what we find is the highest order difference that describes the differences between AD in both of these data sets is a single gene called PKM, or pyruvate kinase. A little bit of background on pyruvate kinase. Some of you cancer people may know pyruvate kinase. It's a hallmark gene in cancer. It's normally part of the glycolytic pathway, so it's obviously a pyruvate kinase. It has 4 isoforms, the 2 principal isoforms. The one that is PKM1, which is the one that's in the cytoplasm. It's a tetramer, and it functions to optimize metabolics for neural metabolism. So it optimizes oxidative phosphorylation, generating pyruvate and using that pyruvate kinase to feed into the oxidative phosphorylation and the electron transport channel. PK2 on the alternatively is a dimer, and it's the difference between these two is a single splice variation. So the PKM2 has an extra exon called exon 10, so you can distinguish between these two. And while the expression of PKM is not different, total difference, there's a clear difference in the ratio of PKM2 to PKM1. Whereas if you look at the pathway for PKM, you have for PKM1, it's glucose to pyruvate, normal glycolysis to citrate, feeding into the Krebs cycle, oxidative phosphorylation, normal strategy of metabolic activity. With the PKM2, it rather functions differently and goes from pyruvate, increasing LKB, as we've shown before, lactate dehydrogenase A, which converts pyruvate to lactate. That lactate then is not contributing as much to oxidative phosphorylation, and the lactate then becomes a toxic element as well. But there's another more important part to this. So let me just, before I get to the more of the function of PKM2, let me just sort of take this one step further and say we went back to ROS map. Now we have 633 transcriptomes, and what we see, and we can now map these 263 to their Brock stage, And we find that PKM1 isoform downregulates with more severe Baroque staging, whereas PKM2 is upregulated. So again, indicating that this PKM2 isoform switch is correlated with diagnosis of Alzheimer's and severity. If we dig down a little bit deeper into this and look at genes that are associated with the PKM2 version, where you have HIF1 alpha activated, STAT3 activated, and glycolysis genes more heavily activated, we see a serious upregulation of these genes at this point. And this is reflected here in this heat map where you have an increase in these genes in neurons. So to confirm whether or not this actually is happening in vivo, this is all RNA-seq data, but if it's really occurring, we can visualize it. We went to the ADRC and got 19 patient brains, and we're able to stain them with PKM2. And what you can see this almost at the light microscopic level here, you can see an increased level of staining of PKM2 in the frontal cortex of 80 patients. And this is their average fluorescence. If we look more at a individual cell level, we're seeing a co-localization of PKM2 with PKM with nu-in. So we're seeing it expressed in neurons, and we're seeing the intensity higher in the nucleus than the cytoplasm. This is a key feature of this that I'm going to expand on in a minute further. Once I convince you that we're seeing this shift of PKM2 from cytoplasmic to nuclear. And this is true for total PKM as well as the enzymatic activity of PKM. We have taken this one step further using mass spec metabolic mapping. So you label carbon 6 with C13, and you follow the glucose flux through its metabolic pathway, and what we see is an increase in lactate, as we predicted to see with RNA and everything else, but now we can actually measure labeled lactate is elevated. So that proves that the cells are shuttling through this alternative pathway that PTA2 forces them through. If you look all in all at this metabolic shift, you see that there's a Warburg-like metabolic profile in AD neurons. Now, Warburg in the 30s came up with this idea that cancer cells shift their metabolic, a somatic cell shifts its metabolic use of oxidative phosphorylation to more of a glycolytic pathway. And in part, the reason why they're doing this is to take advantage of glycolytic phosphate shunt pathway. The phosphate shunt pathway is where you get this switch from glucose into ribose 6-phosphate to make nucleotides, and then further modified to make amino acids, and then also into making lipids. So what cancer cells do is they shift back into glycolysis to have access to the phosphate shunt so they can access to this bulk that they need when they're undergoing cell division. But we think, this is just a theoretical part of it right now, that AD cells are shifting back into this access to get access to this bulk to prepare to repair themselves for the damage that they're going through with this cellular stress. Anyway, these are the metabolic pathways that are being shifted. You can see this increase in this AD on this side and control here. There's sort of a traffic jam occurring at 1,3-bis glycerate, but in the net impact is an increase in lactate in the AD compared to the controls. How am I doing time-wise? Still okay?
Yes. Yes.
Hope I'm not going too fast here.
No.
All right.
Yeah.
Okay, so let's take a look at see what is the molecular difference and cellular difference between PKM1 and 2 that explains and justifies these differences we're seeing. So what happens normally is that PKM2 is kept cytoplasmically where it can provide the pyruvate it needs to feed into the oxidative phosphorylation pathway. Alternatively, the PKM2 is phosphorylated and transferred into the nucleus. There, it co-binds with HIF-alpha and enhances the binding of HIF-alpha, HIF-1 alpha, and STAT3, and induces a severe stress and pro-apoptotic genes, and further amplifies this glycolytic pathway. Now, to prove that in our cells this is happening, we can see that even in our neurons, we're getting a shift of the cytoplasmic PKM2 into the nucleus. And we can look, let me get you out of the way here. We get this downstream target gene, which is H3T11, which is a PKM nuclear target gene. So which in the nucleus, you do see this upregulation, and we see it too. So it's not just transferring this protein to the nucleus, it's upregulating many of these Warburg-like genes. If we then do ATAC-seq on this data and look at the chromatin around these targeted PKM2 targeted genes like HIF-1 alpha, STAT3, CT, and MV1, they all have, these two have significant chromatin opening relative to AD relative controls. No different in this one, but these two clearly are showing open chromatin as well. So there's a increased chromatin accessibility for HIF-1 alpha also as part of the gene program that's involved. Now, one of the hallmarks of this reprogramming is an increase in apoptosis. And however, in culture, INs are we treat our cells very nicely, give them lots of nutriments, and they can stay pretty healthy. They don't undergo apoptosis under normal conditions. But if we downregulate BCL2, for example, with ABT373, then we can see whether or not we can make them vulnerable. And what you do see here is that by treating the AD cells with a BCL2 inhibitor, we get an increase in apoptosis in these cells. Now, cobalt is also or CODO as it's called, and has been found in the chemistry literature to increase metabolic shift. So it's a drug that can mimic the effect of the Warburg effect. So we take wild type INs, convert them into Warburg-like cells. And when we do that, we over-affect our cells with a fusion protein of PKM2 and GFP, and we map it in terms of its localization. And we see that there is an increased nuclear localization of PKM2 when you induce a metabolic shift. Again, confirming that PKM2 nuclear localization is induced by the Warburg effect. When we do this, and then we also treat with ABT 737, we get an increase in apoptosis as well in this separate model of metabolic shift. This is not in INs again. This is just confirming this Warburg-like involvement. And then finally, we can demonstrate even the same phenomena by overexpressing, and the specificity of this with overexpressing PKM2 itself within cells, both increasing apoptosis, but revealing much of the same kind of metabolic shifting in all these pathways that we see. So both mimicking with CODO as well as overexpressing PKM2 induces this Warburg effect, and the same kinds of things we're seeing in the induced Alzheimer's disease neurons. So what can we do? What can we do to protect these cells against that? Well, because this is such a well-known target in cancer, there are drugs being developed for cancer for shifting PKM2 to PKM1. And one of them is a natural product called chiconin. And this wonder drug, forces the dimer PKM2 to heterodimerize. And when it heterodimerizes, the theory goes, it won't be able to get into the nucleus, it'll stay in the cytoplasm, and it won't activate HIF1 alpha or STAT3 and get that whole pathway going. So we did this, treated our cells with Shikonin, and the first thing you can see is this cobalt chloride induced nuclear localization is blocked. So we can prevent the influx of PKM2 to the nucleus, and we can reverse much of the metabolic dysfunction. So our lactate pyruvate ratio is back to normal. This stumbling block here at the 1,3 bis glycerol is not affected. In addition, we can block the apoptotic effect that is normally upregulated in the presence of APT 737 in the AD neurons. And then when we go further to look at RNA-seq on the AD and controls in the presence of Shikonin, whereas normally we see the ADs showing a very nice shift difference from the controls, these arrows indicate the RNA-seq directionality shift that occurs with Shikonin treatment. So Shikonin induces a transcriptome shift in AD and it's back to their more control-like state. Remarkably, when we then go and look at the RNA factors that we found that were downregulated in AD, that is synaptic transmission, synaptic plasticity, long-term potentiation, those are reversed also in part by this treatment with Shikonin. So and something I didn't spend a lot of time with, but I did tell you that with AD compared to the controls, which is this pink, there's sort of a de-differentiation. The cells don't fully mature, but with Shikonin in the same AD patients, we're getting a much more mature phenotype or trajectory of differentiation. So to summarize here at the end, there's a cancer-like PKM2 that it's upregulated. The splice form variation that occurs in AD is causes a metabolic shift and a fate loss. And the cells in AD become vulnerable, resulting in loss of function in terms of metabolic activity and gain of function epigenetically. There's an exchange of profile, and this looks very much like cancer in the sense of the changes that we're seeing. One thing I'd like to point out to you, if you look at this chart here, these are how different cells in the central nervous system use different metabolic activity. So from proliferation, glycolysis, metabolic flexibility, anabolic pathways, neurons are unique in their dependence on oxidative phosphorylation for generating most of their ATP. Other cells share glycolysis and other fates in their ability to generate energy. But this also means that AD neurons cannot easily use glycolysis and the phosphate shunt pathway to develop to generate more nucleotides or amino acids or lipids that they may need in the damaged case. So I think what this tells me is that neurons are particularly vulnerable to damage because they don't have this flexibility in energy metabolism, and they're so dependent on oxidative phosphorylation. They're fine as long as everything is healthy, but as we age, it makes neurons more vulnerable than any of the other cells in the brain. But I'll just say this. Our model, as we look at it right now, only looks at neurons. I've been telling you stories only about neurons. And I think in the next phase, we need to begin to look at more complex systems. Do I have time to tell a little bit more? The organoid story?
Yes, you have 10 minutes more.
Okay. So let's see. Yeah. Yeah, this just sort of emphasizes this, all these different differences between all these cells. Okay. So recently we've developed, so one of the problems with organoids, which we thought would be three-dimensional structures that are more brain-like, but if you look at them very carefully, most organoids are primarily neurons. They're not made up of other cell types. And so, and that's one problem. The other problem is after 3 months, they rot in the middle. So they have they lose oxygen and they lose their nutriments to the core, so they rot. So what we've been able to do is to transplant them at about 2 months of age into an immunocompromised mouse, and they survive there, become vascularized, and you can see blood flow. So here's dextran flowing in blood vessels within a human organoid implanted into a mouse. Now, this is a pretty good model, but what it's missing, it's importing the endothelial cells and the parasites from the mouse in. And you do see some astrocytes sometimes, but they turn out they're all mouse-derived, and so are the microglia. So we wanted to develop a method where we could induce microglia and astrocytes within human astrocytes and human microglia within the organoids. So what we do is we transplant, and we have done that. So we've developed a microglia cultures where we can take human fibroblasts, make microglia, and then we infuse them into the organoids using a very simple method of convection. And then we transplant the organoid into the cavity that we've used in the past. And what we see are multiple human microglia throughout the organoid.
Oh, here goes.
And you can see this complexity. You can see the complexity of these microglia, human microglia in the human organoid, and they continue to mature from 2 months to 6 months in vivo. So here's some branches, but here's multiple branched microglia. And they're making some of the genes that you cannot see in microglia in vitro in 2 dimensions. So P2RY12 and TEM119 are all expressed in these human organoids in vivo. Now, one of the major tests to see whether or not the cells are really functional is to do imaging. So we've collaborated with Axel Nimmerham here at the Salk Institute, and this work was done together with Abed Mansour and Simon Schaefer and Axel Nimmerham, and we used 2-photon imaging of a glass cover slip overlooking the image, and we genetically engineer the microglia so they're red or tomatoes, so we can see them here. Then we image them with time, and what you can see is that the microglia are sensing their environment, just like wild type intact mouse microglia have shown to be done by, in fact, Axel Nimmerham 10 years ago. So to test whether or not they are functioning as microglia, they're sensing the environment in baseline condition, what he can do is he can use a laser beam to kill a neuron here, and then we can watch how these microglia act in response to this response. And as you know, microglia are phagocytic cells. So let's watch what they do. So they target the damaged area and phagocytize this damaged area at a distance, demonstrating that in fact they're functional. And here's the quantitation of this, looking at the process polarization per focal lesion beam, and we see a dramatic increase both 10 minutes and 30 minutes after the lesion. So with that, I'll end in time. Much thanks to Jerome Mertens, collaborator, ex-postdoc collaborator, Larissa, many postdocs and graduate students that have passed in the lab over the years. A lot of people from UCSD there who have helped both with many aspects of this, our funding agencies, and a special thank you to the core facilities at the SALC that allow us to do many of these experiments at a very high level with great instrumentation. With that, I'll end. Thank you.